Hello, everybody, and welcome. Welcome to our second session of the Mini Medical School, The Promise and Power of Genome Engineering. My name is Steph Kennelly, and I'm the program manager at the Center for Genome Engineering at the University of Minnesota. And we're so excited to have you here today. Hopefully, you were able to join us last week. We provided an overview of genome engineering. If you were unable to attend, I would invite you to visit the YouTube channel and view the recording. It provides a lot of really great background. But for today, we're going to jump into our second topic in the series and delve into how genome engineering is offering new hope for people living with rare genetic conditions. So just to remind you of the format, we're going to begin with our panelists providing some slides and information. And at the end, we will have a Q&A session. So you can submit your questions to our panelists at any time using the Q&A box down there, which can be found at the bottom of your Zoom screen. Please note this session is recorded and it will be available starting tomorrow on our YouTube channel at the Office of Academic and Clinical Affairs. We will also send a follow-up email, which will include the link as well. So today we have the privilege of speaking with an amazing panel, four of my most favorite humans that have lived the walk and the talk of rare disease. So we are going to discuss how research is targeting and correcting gene errors at the root cause, opening doors to new treatments that's possible, and we'll also have an amazing perspective from parents and our action happening at the legislature. So first, I'd like to introduce Dr. Joey Skeet. Dr. Skeet is an assistant professor in the Department of Pediatrics, Division of Hematology and Oncology. His research focuses on translational gene and cell therapies for rare pediatric diseases. So Joey, today, can you discuss engineering immune cells and how you use that to treat rare disease?
It would be my honor. Just make sure, yep, I see my voice going and I got the thumbs up over there. So hello, everybody. My name is Joseph Skeet. I'm an assistant professor at the University of Minnesota in the Department of Pediatrics. And I'm going to briefly go over what we define as rare disease here in this field and give you an idea of how big and massive it can be. And then highlight one promising approach that came out of the lab where I did my postdoctoral training, where we are able to engineer primary T cells as a method to provide kind of a living drug and the enzyme replacement therapy that is underpinning that rare disease, or helps alleviate that rare disease. So disclosures, I have no actual or potential conflicts of interest in relation to this program or presentation. And so what I'd like to do now is just kind of start out and center everybody on what we define as a rare disease. So according to the NIH at the Genetic and Rare Disease Information Center, rare disease is defined as a disease that impacts fewer than 200,000 Americans. Currently, and I just downloaded the list today and it's still expanding, there are over 10,000 rare diseases that impact between 25 and 30 million Americans in total. And the cause of many of these rare diseases is unknown, but they're often created by errors in a person's genes or chromosomes that lead to deficiencies or abnormalities within cellular processes, and then eventually lead to other problems during development. As I said, there are many types of rare diseases. These include rare cancers that, the distinct types of cancers that fall below the 200,000 person threshold, such as childhood sarcomas, rhabdomyosarcoma specifically, neurological disorders, conditions that affect the brain, spinal cord, nerves. A category includes motor neuron diseases and rare epilepsies, metabolic disorders, which is something I will be highlighting later on, which is a large group of genetic disorders where the body cannot properly turn food into energy or take out and recycle cellular waste or byproducts during metabolic processes. Immunological and autoimmune disorders, which are diseases affecting the immune system, including primary immune deficiencies where people can't make an immune system, and rare autoimmune diseases where their immune system attacks the body itself. Additionally, there are cardiovascular diseases where there are rare conditions affecting the heart and how the blood vessels form. Hematologic or blood malignancies, which affects the blood and blood-forming organs. Endocrine disorders that affects the endocrine glands, hormones, and adrenal glands, and other certain types of thyroid diseases. I kind of group these two together, musculoskeletal and connective tissue disorders. So disorders that can impact growth and development of bones, joints, muscles, and connective tissues. Connective tissue disorders such as epidermolysis bullosa, which is a devastating blistering disease. In addition, this isn't all of them, but respiratory disorders as well, which are rare diseases of the lungs and respiratory systems, such as CF and idiopathic pulmonary fibrosis. So let's be very clear. The road to diagnosis is often long and challenging, especially in the area of rare disease. And it's typically and historically, this has been because the start of symptoms leads to a first visit that then kind of follows this pathway where people go back, they get their symptoms treated, and eventually they wind up at a specialist, and then another specialist visit, and eventually they get the genetic testing or they find the right specialist that can give them the correct diagnosis or the correct genetic test results. And then even after that point in time, um, most of the rare diseases that occur, you wind up with the support of a rare disease communities focused around that specific disease. And devastatingly, although we have new genomic screening tools and methods that are enabling faster diagnosis and being able to pinpoint what the root cause of a lot of these are and detect them earlier on, there still exists over 95% of these rare diseases do not have an FDA approved therapy to even begin treatment or, correct long-term impacts. But importantly, there are new genome engineering tools that are driving forward both preclinical development in the space of correcting inborn errors at the genomic level, but also, as I'll highlight today, ways where we can engineer novel cell therapies and preclinically test them and give as living drugs. So just to kind of recapitulate who we are at the Center for Genome Engineering, we are genome engineers. And Dr. Largaspato went over this in great detail last week, but just to kind of highlight it, like an automotive engineer that can go in and fix your car, swap out parts, make improvements, we work right on the genetic code. So we change or add DNA code of living cells to change or modify their function. And at a high level, this is done through two primary major methods. One is the transposon-based method, which I'll touch base on at the end of the talk, and then also utilizing and retooling the bacterial defense system, the CRISPR-Cas9 system, where we can directly insert genes, modify genes, or correct things at the individual nucleotide level. But ultimately, genome engineering can be used to add new DNA, correct DNA, and lead to changes in cellular function. Now, going back to one wonderful approach that we're utilizing to treat metabolic rare disease, specifically a lysosomal storage disorder, I will go ahead and kind of give an overview of what these are more in depth so you have an idea and what we're doing at the Center for Genome Engineering to approach and give a living drug to this devastating rare disease. So lysosomal storage disorders involve lysosomes, which are organelles, so little mini organs within the cell that break down cellular waste, debris, and foreign materials. Deficiencies in the ability of lysosomes to break down the trash and the leftover stuff from regular cell processes results in disorder and buildup of that garbage. Within the lysosomal storage field, there's a number of them. So there's more than 50 that have been identified, and they're caused by many different mutations that range in a wide variety of severity. Systemic manifestations and complications occur due to buildup. And one that I will be highlighting today where we are making a cellular therapy is mucopolysaccharidosis type 1, MPS 1, which essentially is a deficiency in the lysosomal enzyme called IDUA. We're not going to go into the long name of this, which is responsible for breaking down a waste sugar called glycosaminoglycans, so GAGs. And this leads to MPS 1 itself, and it actually has a spectrum of severity based on how much of a deficiency you have in the IDUA enzyme. And this disease severity can basically lead from a shortened lifespan of 7 years, but also can have more of an attenuated form that can last all the way into lifehood. The current therapeutic approaches for lysosomal storage disorders have three prongs. The first one is enzyme replacement therapy, and this definitely improves quality of life. However, it's very costly, requires frequent health visits and specialty care, does not improve neurocognitive functions generally, and is, as I said before, incredibly expensive and invasive. There's also an avenue of hematopoietic stem cell transplants, and this is somewhat effective in neurocognitive decline. And ideally, it is carried out within the first 2 years of life, so you can reconstitute essentially a healthy cell population within the patient. But on kind of a downside is you do need a matching donor, and your engraftment rates do vary. And then finally, there's a number of gene therapies that are in clinical trials right now for in vivo delivery, and these include viral vectors. Now, these are once in a lifetime treatments. Not everybody is currently a candidate, and viral vectors bring several risks towards therapy options. And often, your delivery is localized to one organ or region. But all in all, each one of these approaches to treating lysosomal storage disorder relies on this idea of enzyme cross-correction. And that's where if you get a healthy cell or genetically modify a cell that can overproduce the enzyme that the enzyme deficient cell does not have, it can travel over, interact with surface receptors, and be trafficked in and correct the unhealthy cell just by overproducing this. And this is generally the concept of what enzyme replacement therapy is. Now, essentially, the next generation that we're looking at the Center for Genome Engineering is making living micropharmacies. Now, micropharmacies are engineered cells that deliver therapeutic proteins or enzymes directly within the body and exist within the body. So currently there are several efforts out there which include engineered hematopoietic stem cells to produce enzymes that treat lysosomal storage disorders. But importantly, and one of the things we are looking at are T cell-based micropharmacies to deliver therapeutic biologics. Now, T cells or T lymphocytes are a critical type of white blood cell that's essentially at the heart of your adaptive immune system, the part of your immunity that learns and recognizes to fight specific pathogens and transformed cells. Now, these are the immune system's forces that essentially train to recognize self versus non-self, and they develop in an organ called the thymus. This is essentially where they get their T lymphocyte from. Some of these cells go on to become memory T cells, which stick around for years and allow your body to mount much faster and stronger attacks. And we essentially want to take advantage of this long-lived and systemic life of the T cell that travels throughout the body to function as kind of a cell with a second job and pump out a bunch of soluble protein within the body and facilitate that cross correction. I will be highlighting specifically, and everybody can look this up because this is an open access paper, the approach that we utilized as a potential therapy for MPS1. And this came out of the Moriarty and Weber laboratories where I did my postdoctoral training. Now, within this idea of engineering a micropharmacy, the workflow is pretty straightforward. The researchers were able to take blood from a healthy donor, isolate out the memory T cells from the donor, engineer them with CRISPR-Cas9, which I'll give you a quick overview here in just a second. They showed that these T cells were able to produce this cross-corrective IDUA enzyme in a great amount, and that had cross-corrective capabilities on MPS1 deficient cells. And they also tested an MPS1 preclinical mouse model and showed cross-corrective activity there. So the first part was showing that they could engineer IDUA micropharmacies utilizing genome engineering. And to do this, they used the CRISPR-Cas9 system. And all you need to know as a viewer here is they took a genetic code for the IDUA protein, put on a promoter, they packaged it up, and then they isolated memory T cells and delivered Cas9 reagents. Those Cas9 reagents went to a specific site in the genome where it nicked in a double-stranded fashion the DNA, and this template lined up with the DNA here and was inserted. And this facilitated the T cells, so it was able to engineer these T cells, which you're seeing over here, to then overproduce IDUA protein. And they carried out a number of cross-corrective studies where they showed that proteins produced by these cells could correct cells deficient in this IDUA enzyme. But more importantly, what they did was they introduced these cells into mice that already had, or a mouse model of MPS1. So normally these mice do not have any human cells floating around. They engineered cells and introduced them, and they found that they were persistent and they could exist throughout the duration of the study. But more importantly, they found that IDUA was detectable in the blood at near normal levels compared that in mice that received the cell therapy compared to mice that did not have the IDUA therapy. Now, this wound up resulting in a a detectable level of IDUA within tissues throughout the body. So the heart, lung, liver, spleen, kidney, brain, and bone marrow all had detectable levels of the IDUA enzyme, where normally in the MPS1 knockout mice, they did not see that. And then importantly, they were able to see a correction of the glycosaminoglycans, so the GAGs that were found within these tissues, so the deposits of the waste tissue. And that is seen here in the MPS1 knockout mice in pink. But more importantly, when you introduce the cell therapy, there's a dramatic decrease in all tissues that were investigated. So again, I just kind of want to give you a high level conclusion and summary of the talk that I gave so far and the beginning of the opening of our webinar today. The things I want you to take home so far are that rare disease is defined as a condition that impacts less than 200,000 individuals in the United States. Historically, individuals who have a rare disease have a long road to diagnosis and treatments, but we do believe that this is changing with the advent of genome engineering and cell and gene therapies. And importantly, new genome engineering approaches are unlocking novel strategies to treat those diseases, especially in the setting of rare disease treated with enzyme replacement therapies where we can induce cross-correction. So yeah, with that, I do want to acknowledge all of the team that I work with in the Moriarty and Weber lab, and specifically Dr. Kleinbo, whose work that was. And that is my presentation for today.
Thank you so much, Joey. That was really great. As a reminder to everybody, you can please continue to submit your questions down in the Q&A box. And after all the panelists have completed, then we will have an open Q&A time. So next, we are so excited to welcome Dan Bourne. This is a really important perspective, in my opinion, to hear from the parents as well. And Dan's been a huge advocate for us at the center. He's the co-founder of Helen's Pink Sky Foundation. This is a nonprofit that he and his wife established in honor of his daughter, Helen, who was diagnosed with Batten's disease. Through the foundation, Dan and his family are committed to accelerating research, raising awareness, and building a community of support for children and families that are impacted by Batten's disease. So Dan, welcome. And can you please discuss your experience as a parent of a child living with a rare disease?
Absolutely. Good evening, everybody. My name is Dan Bourne. As Steph said, I'm one of the co-founders of Helen's Pink Sky Foundation. And I'm just going to speak a little bit today about our journey from diagnosis with our daughter into action with rare diseases. I hate asking this question, but my slides are showing up, right? So good. All right. So I'll take you through just a little bit of our story. I put some pictures in here of Helen and her older brother George, because I think it just kind of helps you visualize what it is that we're going through. So Helen was born on May 31st, 2021. She was happy, she was healthy, and she kind of completed our family. She has an older brother George, who you'll see in the picture on the left. He's 2 years older, and he's Helen's protector. He is Helen's best friend, and just absolutely adores her. We lived in Colorado at the time, and moved back here, or moved to Minnesota right about the time Helen turned one. So the next picture you see here is from Helen's first birthday, and the third picture is from Helen's second birthday. And then the last picture with the cake you see is from Helen's third birthday. And actually, as I was making these slides, going through my camera roll last night, and that was the last picture that I have of Helen. Excuse me. Before things just kind of started to go sideways. So let me take a drink of water and get myself under control here. So take you through the diagnosis. Like I said, Helen was happy and healthy and just a beautiful little girl until she turned 3 when things started happening. And I'll just walk you through a little bit about what we experienced. On June 9th of 2024, so June last summer, 9 days after Helen's birthday, she experienced her first seizure, completely out of nowhere. There was no signs leading up to it. We called 911, not knowing what was happening, and an ambulance came, took us down to Children's downtown, and they said, every kid gets a blip. Brains are developing, and hopefully this is nothing to worry about. They sent us home, and we kind of went on with our life and chalked it up to a scary incident. About a month and a half later, Helen experienced her second seizure um, towards the end of July. And again, we called 911. Ambulance came and took us downtown to Children's. And she went through some testing at that point, um, and diagnosed Helen with epilepsy, which at the time was devastating for our family. It was scary. All of a sudden, things are a little bit unknown. And as I sit here today, I'd be very grateful um, if epilepsy was the diagnosis we were still living with. Um, Towards the end of 2024, call it early winter, we started noticing additional symptoms. And Helen would have these, you'd be talking to her, and she'd have these eye flutters. Her head would kind of, start dipping. She would start falling, just walking across just a wood floor. She would trip and fall. She would have these tremors. She was, struggling all of a sudden with using utensils to eat. Her speech was declining, and things were just changing. And it didn't sit well with us, and we figured we needed to go, get some answers. So we ended up going down to Mayo. Um, at the beginning of 2025, January, February timeframe, and talked with them and decided that it was probably best um, to go through and do a 24-hour EEG to test Helen's brain activity, and also do genetic testing at that point. And the reason for the genetic testing, something we'd previously declined, was with the thought that if they could figure out exactly um, what was happening with Helen, they would be able to tailor her medication best to control all types of seizures. It turned out, I'll go back to the eye flutters and falling and tremors. It turned out that was at the time it was diagnosed as a different type of epilepsy, and so she's put on an additional medication, and but it just wasn't working. So March 3rd, that's when we went down to Mayo and spent 24 hours there. Did the genetic testing, both myself and my wife, Helen's mom, did genetic testing as well, so they could triangulate all information. And we left that appointment. I still thought our life would remain the same, and we'd get through this. And they told us, results will be back in 3 to 4 weeks, and we'll call you when we know anything. And it was a week later, on March 10th, that I received a message from Mayo saying that they'd received some results back, and they needed to talk, and that they would make themselves available that day. It was important that we spoke. And so my wife and I gathered at the table, and it was a Zoom meeting. We signed on to Zoom, and um, there was a couple doctors on the screen that you could just see in their face that it was going to be an unpleasant conversation. And they proceeded to tell us that the results were back, and um, that it was very bad. And they introduced us at that time to something that we had never heard of, and I'm assuming many of you have never heard of it either. It's called Batten disease. And Batten disease is really an umbrella of these lysosomal storage disease in this family. Specifically, Helen has CLN2, which is a late infantile Batten disease. There's CLN1, CLN2, CLN3, and so on. I believe there's like 12 of them. Um, and CLN2 Batten disease usually presents itself in the form of seizures uh, from the ages of 2 to 4. And Batten disease is a neurodegenerative disease um, that there's no cure for today. Um, the only thing that can be really done to slow progression is something that's called an enzyme replacement therapy, which Dr. Skeet touched on just a little bit there. So, exactly a week after receiving this diagnosis, Helen underwent brain surgery to um, to insert an Ommaya reservoir, which sits in the front of her head. And every 2 weeks we go down to Mayo. It's a uh, full day therapy, and they give Helen the enzyme that she lacks. So CLN2 is, I'll go back to kind of what it is real quick. Um, Helen does not have an enzyme that is responsible for, it's called TPP1, and she lacks that enzyme that is responsible for breaking down waste within cells. And so the waste builds up in Helen's cells, and those cells die off. And so, as I said, it begins with seizures. It But the progression of it is um, it's really bad. Um, the doctors told us that when they introduced us to this, they told us that Helen would lose her ability to talk. She would lose her ability to walk. Um, she would lose her ability to eat. She would go blind. She would become bedridden, and and then that would be the end. Um, and there's nothing that we can do about it. There's no cure. There's just this enzyme replacement therapy, which buys you a little bit of time that slows the progression down a bit. Um, and that's really where Helen's Pink Sky Foundation was formed, was um, out of being a parent that loves your kids so much and saying, this is not acceptable. This is not something that I can just say okay to, and that I can live with. And I guess we're fortunate. I'll go to the next slide here. We're fortunate to have a family that is incredibly supportive and feels the same way that we do, that this is not something we can accept. And so we decided that we were gonna make progress, that we were going to find a cure for this. And so we started Helen's Pink Sky Foundation on April 9th. So very shortly after this diagnosis, about a month later. And our mission is to raise money and fund research and find a cure for CLN2 Batten disease for Helen, but also for all other families that are dealing with this disease and will deal with this disease in the future. There's about 200, to give you an idea of how rare it is. There's about 200 kids that are living with CLN2 Batten disease today. We began fundraising immediately. I noted the IRS letter of determination in case anybody is going through something similar. This was one place where we kind of hit a roadblock that we just couldn't get over. We had lined up different fundraisers, and we were getting donations, and things were in motion, but we didn't have our IRS letter of determination, and so we were unable to turn that over to folks for tax benefits, for write-offs. It limited the institutions that would do fundraising for us. It limited the ways that people could donate. Um, and so I just wanted to call that out really quick as kind of something to be cautious of and plan ahead for. We actually had a very large event, which I'll talk about here in a minute, that we were able to get the letter of determination like the day of. We were right by the. It was scary that thinking that we had this huge event with I'll call it a national stage, and we were missing one core piece, but we knew that time is not on our side. Um, the doctors say that, the goal is that Helen would make it to 10. She's 4.5 now. And so we don't have a lot of time. We had to get to work. Um, I've never turned down since this diagnosis. I don't think I think I can confidently say this. I haven't turned down any opportunities to speak, to get the word out, to raise awareness um, for this disease and for Helen. And I had the opportunity to attend. It was virtual, um, a rare disease day put on by the state of Minnesota, and you're actually going to hear from Erica Barnes here in just a little bit. It was her group that put it on, and I had the opportunity to during Q&A at the end where I spoke up and I just said I want to talk through my family and what it is that we're going through, and I introduced them to the story and um, what we were experiencing with Helen and what our plans were. And I'm fortunate that both Steph Kennelly and Dr. Skeet were on that call, and they reached out afterwards and said we'd like to talk. And we went into the lab to meet with the team. They had never done anything with Batten disease, but it was very apparent that they'd done a lot of research and they believed that they had something uh, that was worth chasing down, that they thought might be able to um, to solve this disease. And Dr. Skeet touched on it before. It's T cell therapy, and basically it would create micropharmacies of this TPP1 enzyme in Helen that would transfer from cell to cell and give her the ability to break down the waste. Um, Really from the day after diagnosis, maybe it took a couple days for me to figure out what just happened and get my you know be able to think. But I started doing research, and um, I would read papers on Batten disease, basically anything that was out there from a research paper perspective. And at the end of every research paper that's listed, all the folks that participated in writing that paper and the research and whatnot. And I started reaching out to with help of my family, reaching out to everybody that was listed on those papers. And I'll say I was quite surprised at how many responses I got. Either folks left a voicemail and they called me right back, emails they emailed back. Um, People were there to help and were wanted to give every little nugget of advice or insight that they had. And we talked to 50 to 100 different doctors and researchers all over the world in the month after diagnosis. And we were able to kind of identify different groups that we felt would be uh, maybe worth sponsoring research at. And after meeting um, University of Minnesota, Dr. Skeet and Moriarty and their teams, we felt that they were one that deserved additional due diligence. And so what we did is we went through and we gathered proposals from everybody that we talked to that we felt would be worthwhile. And in those proposals, we started looking at what is the cost of this going to be. Many of them were in the $5 to $10 million range over a number of years. So we're not talking we're talking incredible expenses here. Um, and they also took time. And at this point, we have funded research at the University of Minnesota Moriarty Weber Lab. We've funded research with a biopharma company out east called Lattice Bio, and we're also funding some research at the Children's Hospital of Philadelphia. So we've issued research grants that total $1 million at this point. And those are really for kind of the first phase of the research. It'll go into phase 2 and 3, and once we prove phase 1 successful. So it'd be time for questions at the end if anybody has any questions about that. But I just wanted to show some of the positives that we're doing, and we've been able to do in a very, very short six months amount of time. I often say that we're very fortunate during a very, very unfortunate situation. With the letter of determination, that event that we had, we were fortunate enough to have a national audience with a special that ran on Helen during the Indianapolis 500 coverage. And this picture here is Helen's big brother George. We had Helen's Pink Lemonade stand at the Indianapolis 500, the Motor Speedway. And there's George out drumming up interest and education and donations. We've had Archbishop Hebda at Helen's school. I would have dressed her more appropriately had I known this was happening, but it was just another school day in my mind. But he blessed Helen during Helen's fourth birthday on May 31st. That weekend, there was hundreds of Helen's Pink Lemonade stands that friends and friends of friends and friends of friends of friends hosted throughout the country. And this picture in the middle here is one of the ones in Excelsior. This was on the last day of my son and daughter's school. They asked everybody to wear pink to generate awareness, and this was a picture of their of school wide, and everybody supporting Helen in pink. If you look real close, this is Helen sitting on one of her teacher's laps down here at the bottom. This is a picture here of when we made our first donation to the University of Minnesota Foundation for the research that Dr. Skeet and his team is doing. This is on a visit to one of our labs. I'll just give a real quick plug to U of M, Dr. Skeet and Dr. Moriarty and their supporting cast. It was apparent from that, since that very first visit, from my first phone call with Steph, that they cared deeply for us and for Helen, and they would do anything possible to help us. And I can never thank them enough for that. It means so much. Dr. Skeet brought his daughter, who's also 4, to Helen's 4th birthday party, which was incredibly meaningful. And the support and love that we've received from the University of Minnesota has been nothing short of incredible. And then this one is just actually last weekend. My wife and I had the opportunity to go to the F1 race in Austin for the Grand Prix. And if you look real close here, this is Helen's logo on the front of the Alpine cars. And the team was absolutely amazing. They all wore Helen's Pink Sky bracelets throughout the weekend, and did a lot on their social media to continue to generate awareness for Helen's Pink Sky Foundation. I'm probably over. I tried to start my clock and it went off. So this is my last bit. Our first annual Helen's Pink Sky Foundation Gala is this Saturday, October 25th. Tickets have closed, but there will be a second annual. If you'd like to learn more about the foundation and about our story, you can go to www.hopeforhelen.org. And there is something that is live that you can get involved in immediately, which is the auction that is going with the gala. It closes 9 p.m. on Saturday, and there are some absolutely amazing giveaways on there from different experiences to sports memorabilia, a Taylor Swift autographed guitar. Um, we've been able to compile through generous donors and through network an incredibly impressive auction. So I would love if you went to that site and checked it out. And if there's something on there that you find yourself interested in, place a bid. But that's what I have for now. I appreciate you guys giving me the time to speak and introduce the foundation.
Dan, thank you so much. I feel like I just need to take a second to take a breath there. That was really beautiful, and I know it was echoed in the chat too, just how appreciative everybody was of the courage and vulnerability and love that you shared. And I know that that message resonated with me and resonated with a lot of the people listening. So just thank you so much for showing up and sharing that story. As a reminder to everybody, you can continue to put your questions down in the Q&A box, and we will have an opportunity to answer those at the end as well. So now it is my pleasure to bring on this dynamic duo, and I know that Dan alluded to it, but it is kind of fun because our whole crew, Joey and Erica and Paul and Dan and myself, we all were at that Rare Disease Council meeting that Dan mentioned. And so we're really fortunate to have Dr. Paul Orchard and Erica Barnes. Dr. Orchard is the medical director of the Inherited Metabolic Storage Disease Program and a professor at the Department of Pediatrics. He does clinical research and is evolving new and combination therapies to decrease toxicity and improve outcomes with a particular interest in the use of gene therapy approaches. And Erica is the executive director of the Minnesota Rare Disease Advisory Council. She has over a decade of advocacy and nonprofit leadership experience, and she co-founded with her husband, Chloe's Fight for Rare Disease Foundation, following the death of her daughter. And together they bring such an amazing and unique skill set. So Paul and Erica, can you please share with us some of the opportunities and challenges in implementing gene therapy in rare disease?
Very
Thanks,
good.
Steph. I think, Paul, you're going to go first.
If that's okay.
And I'll try not to jump in and correct you.
That's okay. You're certainly welcome to do that. So, Steph, thanks for the introduction. I'm assuming people can see the slides, right? So, Joe, I appreciate the presentation you did earlier. I'm going to rely on some of that as we kind of move forward here. And Dan, that was a remarkable presentation. I think really put things in perspective in terms of what some of these diseases are and how they affect the families. Yeah, and go ahead and move it forward. So disclosures for the next slide, and then I wanted to talk a little bit about. Keep going. Yeah, so I mean, why would we talk about using cell and gene therapy for treating rare disorders? You can move that again. Go ahead. So Joey alluded to this, but this is the original paper that talked about cross-correction. So cross-correction essentially means if you have normal cells that can provide enzyme to cells that cannot, they have the ability to correct these cells. So what we see with the red arrow here is some of these cells that are lysosomal disorders accumulate substrate, which is essentially storage material that isn't supposed to be there. The cells are supposed to be able to eliminate that, and then you can advance that. If you put it in with normal cells in the same tissue culture, that accumulation of substrate disappears. So this is really the basis for why we initially launched into using blood stem cell transplant for patients that have for rare disorders. And this goes back actually to the 80s. Next slide. And then you can kind of advance this here. So enzyme that's produced, that's supposed to go to the lysosome, you can go ahead and advance that if you like, picks up a tag. I should have, yeah, there we go, thanks. So they pick up a tag, it's mannose-6-phosphate, that tells the cells that this is a lysosomal enzyme. You can advance it again. There's a receptor within the cell that will then take that into the lysosome. So that's the way these lysosomal enzymes get to the lysosome. You can advance it again. Importantly, though, if you have enzyme present in the environment, there's also receptors on the cell surface, and advance it one more time there, that can recognize that enzyme present in the environment next to the cell, and one more time there, and then that enzyme then makes its way to the lysosome. So this is the mechanism by which, if you're providing enzymes such as the pharmaceutical-based enzyme, that it can help treat these disorders. And this is how the cellular therapy essentially works as well. Next. So again, we started doing blood stem cell transplant for these lysosomal disorders. Essentially, there's a donor cell, so we have to find a good donor for the patients. The donor cells are then making enzyme, and you can advance that again. So that enzyme is present in the environment one more time, and that enzyme can be picked up and utilized by the cell. So one of the issues that we have with treating these kinds of diseases is if we provide enzyme in the blood, it may not make its way into the brain very well. There is a blood-brain barrier that keeps out large molecules, and that can be get in the way, essentially, of treating patients. And so, as Dan had mentioned, some of these approaches are delivering enzyme directly into the brain, into the ventricle and spinal fluid of the brain to help treat them, because if we infuse enzyme intravenously, it doesn't work very well. So we can advance it again. So if we have cells within the central nervous system for some of these disorders, like Batten's, for instance, one more time, that some cells from the donor will actually make their way into the brain and become cells within the brain, like a microglial type cell. Those cells, and again, you can advance it, those cells then can provide enzyme within the central nervous system and serve as a source of enzyme to help treat the disease. Next. So we've been doing allogeneic transplant again for decades for these types of disorders. Transplant has gotten better, the mortality's improved, but it's still at the 10-15% range, which obviously is a major drawback to this. And if we don't have a good matched donor, then the risks just go up from there. And one of the other things that, and Joey kind of alluded to this as well too, is that there's no way to increase how much enzyme is delivered by the donor-derived cells. So whatever cells those cells are making is whatever those cells are making, where there's no way to turn up the gain, for instance, and have them make more enzyme. If we think the amount of enzyme delivered is important in treating these disorders, and that's essentially the thought. Next. So I want to focus on metachromatic leukodystrophy, which is another lysosomal neurologic enzyme deficiency, because we don't have time to go through a lot of disorders, but I'll refer back to a couple different approaches at the end. Next. So metachromatic leukodystrophy is a demyelinating disease. The myelin is the white part of the brain. We talk about leukodystrophies. Leuko means white, so these are areas that affect the white matter as opposed to the gray matter of the brain. This is aerosulfatase A, which is one of the lysosomal enzymes. It's about 1 in 40,000 kids are affected. Because there's about 4 million kids born in the US a year, that'd be like 100 kids that are going to have this on a yearly basis. The stuff that you can't break down and accumulates because you don't have ARSA activity are the sulfatides. That damages the myelin, both in the brain and the peripheral nerves. It's a progressive disorder and is lethal. Next. So we've probably done as many bone marrow transplants for this disease as any place in the country, if not the world. It doesn't work very well. Mortality rate still has been high. A lot of that we think is because we pick up these kids too late to make much of a difference. And it just does not seem to be sufficient in terms of delivering the amount of enzyme that we need to really impact the disease. Next. So this is a patient that I took care of for years that had MLD. She had a transplant done, but even though it slowed the process of the disease, she continued to lose ground. She, towards the end, was essentially almost unable to move. She was on a ventilator at home. Her mother took care of her. It's a very intensive process. She was fed by a G tube, for instance. Obviously, the family gave us approval to show this, but I just wanted to get across to people that these diseases are awful. Next. So this is the gene therapy approach that was developed for this disease. So rather than using somebody else's blood stem cells, essentially this is using the patient's own blood stem cells. So the process is we collect the blood stem cells. Those are CD34 is one of the markers for stem cells. We collect the cells from the kids. There's a virus that we use that has the aryl A gene in it. You can transduce, which is the terminology for infecting these cells, and create or place a normal copy of the aryl A gene in the stem cells. The kids still need to get some chemotherapy to eliminate the cells that aren't producing enzyme, and then they're infused with their own cells as opposed to somebody else's. So those cells can then circulate through the body and make their way into the brain, and then we can see how those kids do. I think there's one more advancement here. And we can talk about this at the end if people want to, but this now got licensed in March 18th, I think, of 24. But this is the cost. It's like $4.25 million for a cell product. Next. These are some of the data from the initial studies that were done. So they compared the kids that were treated with gene therapy to the kids that were untreated. So this is a natural history population. And these are essentially survival curves. And you can see what the difference is in terms of the patients that received gene therapy versus those that did not. Next. And this looks actually at motor function. So how well the kids can walk, for instance, ambulate. And again, the late infantile form is the form that is most severe and shows up earlier, and the juvenile form shows up a little later. But for both groups of or types of disease, you can see there's a dramatic difference in how these kids do if they get this gene therapy. Next. So there are now, I think there may be 6 or 7 sites that are available to offer this, but there's a very rare disease. There's going to be specialized centers that are going to be doing this. So it's not going to be readily available in each state for kids that are diagnosed with MLD. So we were the first site to open. There have been 11 kids treated with actually the 11th is in the hospital now, but we've done 9 out of the 11. Next. And I just wanted to show this is the chemotherapy associated with the standard bone marrow transplant that we've been using for years. Obviously, won't be a test on this, but obviously it's kind of complex in terms of how many agents that are required to do the bone marrow transplants. Next. And this is what we do for the gene therapy patients. So it's much simpler. It's much less toxic. The kids are out of the hospital faster. The family gets to take the kids home quicker. It's clearly an easier process than what we've been doing historically. Next. I just wanted to touch at the end about a couple other different approaches for gene therapy. So Krabbe is, again, another one of these neurologic disorders that is rapidly progressive. There's animal models of Krabbe, and people determined that if you use bone marrow transplant and give the gene therapy, the patients or the animals did better than if you did one or the other. And so essentially that protocol was set up so that when the kids are diagnosed, they would go to bone marrow transplant, and then as they're recovering from the transplant, they would have an infusion of the virus. So in this case, the virus is adeno-associated virus, or AAV, and it's different than we were talking about before because this virus actually is infused intravenously, as opposed to what we were doing before, where we were taking the cells out and engineering them in the laboratory. But again, the idea is to provide a lot of enzyme present that we can't achieve with transplant alone, and then to follow how those kids are going to do. Next. This is another approach for gene therapy for neurologic disorders. So, again, as it's difficult to deliver the enzyme into the central nervous system in sufficient amounts to be able to impact the disease. So here, rather than giving the virus intravenously, you're giving it into the spinal fluid at the back of the brain in an area called the cisterna magna. Next. And the idea is that the spinal fluid that circulates through the brain is going to, and one more time, I think, provide the enzyme into the central nervous system that you need to be able to effectively treat this. So again, this is a different approach, again, with the AAV-based vector. Next. And I think this is the final approach that I was going to mention. And so this is transposon based, as Joy was talking about before. So rather than using a virus, this is essentially using nucleic acids to deliver the important gene here. And so essentially that DNA is administered into the cells with electroporation. Essentially, you pass a current over the cells, and they can actually take up the transposons, and then we give those cells back to the kid. And in this approach, we're actually not using chemotherapy, but relying on these cells to be able to make sufficient enzyme that we can improve their outcomes without having used the chemotherapy. Next. So just in conclusion here, so there's two big pieces that we think about when we're talking about therapy. One is safety and one is efficacy. I think it's absolutely clear for the types of gene therapy we've been talking about, safety is substantially better around the time of comparing it to transplant. It's again, 10 or 15% of the kids may die going through a standard transplant. It's going to be like 1%, if not lower than that, as we use these types of gene therapies. I just wanted to mention that in one of the other disorders, adrenoleukodystrophy or ALD, the way that virus was put together was problematic. And some of these kids have developed actually leukemia. So there's obviously important lessons there. But we haven't seen that in other disorders. It was primarily that virus made for that disease. And in terms of efficacy, there are some patients that may not respond, for instance, but clearly for MLD, as one of these disorders, we get much better outcomes, it seems, than we can achieve with transplant. There's other diseases like Hurler that we're looking at in terms of these types of things. But the costs are substantial, and there are issues in terms of how we're going to be able to deliver this to the kids that need it. I think there's one more slide. So in terms of the gene therapy piece, what are the potential obstacles to the gene therapy? So if you're trying to provide an enzyme and you can have certain types of cells deliver enzyme that's going to be sufficient to all the cells in the body, that's more straightforward. If you're trying to treat a disease where you need to get that gene into every cell in the body, we don't know how to do that yet. One of the issues too is the clinical trial design is difficult. These are rare diseases. There's a fair amount of variation from one patient to another. Sometimes the diseases, it may take an extended amount of time to find out how much impact you're actually making for these types of disorders. And some of these trials cost hundreds of millions of dollars and can take, 10 years to do. So there's substantial issues associated with these things. And so that kind of leads to the whole, question about industry and whether industry is going to be interested in even trying to go there for a lot of these disorders. I mean, there's very few patients trying to get a return on investment, and a business model is difficult. And so I think this is an obstacle in terms of being able to offer gene therapy to a lot of the disorders, is it's just financially not viable. And trying to do things in an academic environment may be part of the answer, but we don't know how to come up with sufficient funds to be able to do some of these things. So There's a lot of discussion about how that might work. And just at the end, I mentioned that some of the biotech companies that have been in the field have decided to pull out. And some of the therapies that we think may have had some real promise are essentially not available now because the companies decided that it wasn't worth continuing. So I think that's the end for me. I'll turn it over to Erica, but that's the hospital. But and then at the end, if there's any questions, I'm more than happy to answer whatever I can answer.
Thank you, Paul. And I am going to pull up my slides quickly, and I won't spend too much time because I know we want to get to the Q&A time. But when Paul and I were talking about sort of what we would present, I think it's really great that I went last because I want to talk a little bit about sort of the trajectory after some of these diseases actually come to fruition, and we actually do see some progress and some promise. What happens after that? Can you all see my slide? Okay. So I want to talk a little bit about moving these available treatments to accessible treatments very quickly. I don't have any disclosures. So my daughter actually passed away from MLD. That's how I got connected with Paul. I've known Paul for years. And it's just been extraordinary to watch the progress. And I just have to echo what Dan said, just the researchers and the clinicians at the University of Minnesota and the dedication that they have to advancing science. And sometimes I always say, sometimes you meet researchers that sort of theoretically want to advance improvement for care for, humanity, but the researchers that I've been blessed to get to know want to do it for individual patients as well. And so it's a really beautiful thing to see. But I just want to really call out that I know my patient community, we always thought, wow, if we could just get an available treatment, an effective and an available treatment through the FDA, everything's going to be great. But I think now in terms of the work I do on the advisory council, I am driven by the question in my work, what good is it if we have all the cutting edge research and patients cannot get that? So we don't get credit as society if we fund and we have innovative technology, if we can't figure out a way to make sure it gets to children like Helen, and we can't make our ecosystem around these technologies such that these children can access them. So I just want to spend a little bit of time talking about that. And then I really want to get to the questions. So the availability of cell and gene therapy has been really exciting. We've seen a steady increase in funding and investment over the last decade or so. We've seen a focus on better infrastructure for regulatory flexibility. The FDA has invested in their infrastructure so that they can have better regulatory pathways that fit these diseases. As Paul said, we have a long way to go, absolutely, but at least there's been movement. And then we have seen some FDA-approved treatments becoming available for diseases like sickle cell and MLD and ALD. So it's exciting. And it's certainly over the last 15 years. So Chloe passed away in 2010, and I never would have imagined the progress that we've been able to make. So it's been exciting. At the same time, access to these therapies has been very concerning. We've seen kind of a rise in barriers to being able to access these drugs. The irony of the fact that sometimes what we see is these flexible regulatory pathways, like the accelerated approval pathways. The FDA will exercise all this flexibility, and then we see some barriers put up, whether it be drug formularies or prior authorizations. So we're really seeing some of those barriers. One of the big barriers we're seeing is a lack of clarity relative to Medicaid's ability to reimburse for these. As Paul said, they're incredibly expensive. They're a one-time upfront cost that the promise of the therapies is that they'll avert cost in the long run, but there's challenges to setting up our reimbursement structures. And that affects our ability to scale these to be able to reach all the kids and across these diseases as well. So we have to be thinking about sustainability. We have a task force on the Rare Disease Advisory Council. Paul is part of it. And we've identified a number of challenges specifically for Minnesota in being able to deliver these. We had to do some work on some legislation. to be able to make it possible for the hospital to get reimbursed for these products. We know that value-based arrangements with manufacturers to sort of track those outcomes are very complex. And again, as I said, the high upfront cost means we have to really commit as an ecosystem to risk sharing as we get these therapies off the ground. So people need to come together, and it can be challenging. Thus, the reason that we in 2019 established the Minnesota Rare Disease Advisory Council. So the Minnesota Rare Disease Advisory Council is an executive branch state agency. We actually started out at the University of Minnesota, and I always say it was kind of like we were a little seed that got planted, and when we grew to the point where we could be our own state agency, we moved over there. So we've been a state agency since 2022, and we've been really working to try to address some of these policy issues. I just wanted to quickly mention a few actions that we've taken to try to align that ecosystem, the reimbursement and delivery ecosystem, with cell and gene therapy. So we've passed a number of pieces of legislation that I'm very proud of. We've really tried to, some of the legislation I put on the slide is directly related to the delivery of cell and gene therapy. So working on a bill that would allow the hospital to get reimbursed separately for this product from the clinical delivery. And we've also worked on legislation to make sure we can diagnose these children faster. So better access to rapid whole genome sequencing, really working on our newborn screening program as well. And then we've worked on some legislation to make sure that patients with rare diseases can access specialists. So that's the work that we've done. We work in collaboration and strive to build partnerships with our Department of Human Services, our Medicaid department, our Attorney General's office, so that we can take all of these amazing treatments and make sure that they get delivered. So that is my presentation. And thank you all so much for letting me share the work we're doing at the council.
Well, thank you so much to all our panelists. That was fantastic. Now we will move on to questions submitted by the attendees. As a reminder, you can still submit your questions to the panelists using the Q&A box. And we will try to get to as many as possible and follow up with an email. So we're going to start first by asking a question to Joey. Joey, you had talked in your presentation about micropharmacies. Can you describe a little bit how those are different than some of the other traditional therapies?
Yeah, so in the presentation, basically, I had showed that we have several approaches, and Paul reiterated we have several approaches in the cell and gene therapy space for reintroducing genes or overexpression of constructs for loss and replacement of these enzymes. Ideally with a living micropharmacy, so a T-cell product that is basically circulating throughout and living long term, you have a constitutive overexpression. So you're continually getting doses versus like an enzyme replacement therapy where you go through ebbs and flows of infusion, valleys of the available enzyme that is circulating. And so ideally what we're trying to do is introduce a modality of treatment where we are providing a continuous supply of the enzyme in question throughout the body. So the short version.
Fantastic. Thank you. Dan, a question for you. As you reflect on your journey thus far with Helen, do you have anything you'd like to share about how you were supported or kept informed by the medical team?
I think it goes back to advocacy. If I'm not going to advocate for my daughter, then who is? Um, in one sense, the doctors are employees of ours, if you will, in my mind. And so I make asks and I ensure that those asks are followed through with. One thing I have done is Helen is the only that we're aware of the only child with CLN2 Batten disease in the state of Minnesota. And when you look at her care team is down at Mayo, you look at an institution like Mayo, and Helen is the only child that is, that is under their care with CLN2 as well. And so it's a bit of a learning curve for everybody. And I've been able to connect Helen's doctor, or care team, with researchers and other physicians that we've connected with, again, across the world, to make sure that they're informed at Mayo, in the best way possible, and kind of triangulating all information that we're uncovering, in real time. So I think a lot of it comes back to, advocacy and, for Helen.
Thank you so much. Paul, a question for you. That slide in your presentation about with the states and highlighting Minnesota was so exciting, because I think that a lot of people don't realize that we really are leaders in gene and cell therapy here in Minnesota. We're kind of a humble people, so it's good to talk ourselves up a little bit. How does your team decide which rare diseases get attention, or which rare disease you'll focus on?
Well, we've been a center that's been interested in these kinds of diseases, going back again decades, actually generations, really now. So, I mean, one of the things I don't put viruses together like Joey does or anything. Way back when I did a little of that, but I stay out of the labs these days. So my role is kind of more on the delivery side, and a lot of these things are coming through industry. And so being aware of what diseases we have expertise in and what companies are moving things forward that we can then apply to these diseases is really part of what I see my role at now. So, we institute the clinical trials, and we've done very well in enrolling patients. I know that sickle cell was talked about a fair amount at the last meeting, and we are one of the largest centers in the country enrolling in the sickle cell gene therapy studies, because we are interested in trying to be a point institution for doing these things. So, yeah, I mean, there's thousands of rare diseases, so it's very difficult to be able to offer something to all of them. But for the ones that we have interest and expertise in, those are the ones that we're focused on for now.
Thank you. Erica, the slide you had showed that there's some bills that you're working on that have House file numbers, Senate file numbers. There were some questions on what people on this call or other advocates can do to support some of that legislation. Do you have any kind of specific tasks that we as the public could do to support the work that you're doing?
Yeah, thank you so much for that question. Absolutely. It is really true that so I began my work in advocacy. Truly, I had this idea that we needed a council, and I had never contacted a legislator before. And I truly went up and just told my story about my daughter to my own legislator, and why rare diseases mattered and why we needed to prioritize them. So I think it is truly true, especially at the state level, that these legislators care and they want to hear from their constituents. So I think that the starting point is find out who your state representative is, and send them an email, give them a call, set up an appointment with them, and just let them know that rare isn't rare. I think that was the first thing that we really had to work on to help the legislature understand that, yes, an individual disease might be rare, but collectively with 10,000, they affect a significant amount of Minnesotans. So telling your story. And then, yes, we're currently working through our policy platform for this coming session. So I would love anyone who wants to be interested in helping us go up to the Capitol and talk to legislators. But starting with your own legislator, if you're an advocate, telling your story, if you're a researcher and a clinician, saying why it's important to you, it's very meaningful. I always say too that the legislators hear from professional, government relations people every day, all day, and that's wonderful. That's what they do for a job, but they really love to hear from actual people that don't do it for a living and are really coming with their story.
Is there somewhere that people could go to get more information on those bills that you have drafted?
Yes, so we will. So the ones that I showed that are on our website right now are what we've worked on in the past. When we get closer to legislative session, we will put up what we're working on, um, and so yeah, that information will be up. We're actually also working on getting a form on our website where people can say what if you have a rare disease or if you're a researcher, what barriers you're facing, so that we sort of have a handle on what we should be working on. That form, if it's not live, it will be live as soon as we can figure out the back end technology.
Fantastic. And maybe we can send that in the follow-up email too for people as well. Next question for Joey. Joey, how are you using AI or kind of with this new supercomputing? Has that had an impact in your research at all?
It has a impact on some of the design implementations we're coming up with for delivering certain cell therapies and formulations. I know it's a little bit vague, but it's a tool that largely relies on data sets that are known and defined. And a lot of data sets in the scientific community are separated out in different formats and in graphical formats. And there's not a lot of raw data yet to build a language model upon to interpolate patterns, et cetera. So it's useful in some aspects of formulations, but it has yet to be fleshed out as a truly very powerful tool in the unknown spaces. Like it's able to refine known things at this point and come up with kind of avenues in that area, but in the very new space, it's not quite there.
Great, thank you. Dan, I know you alluded to this a little bit that, Helen is the only Minnesotan with this CLN2, but you have made connections. Families that are maybe on here that do have rare diseases, do you have any resources or recommendations for families to find support or plug into, that really unique community that you and your family have found helpful?
Yeah, one place that we started right away was if there's like a national organization that whether they financially help, a lot of times they have connections that for different researchers and just information that's very helpful. So for us, it was the Batten Disease Support and Research Association, BDSRA. We got connected with them very, very quickly, like within a day or two, um, and they were able to help connect us with different families that are living in the same situation that we are. We're able to answer questions for us in a very scary, confusing time. I think that's a good place to start is with kind of a national organization that has more information than you do as an individual.
That's a great tip. Thank you. So, Paul, we were talking a little bit about biotech, and I know there's concerns that it's maybe not profitable for them to want to fund these types of therapies. Where do you see if you could dream up a scenario where the university and industry could partner to really have an impact in this? What would that look like for you?
Well, so this is a great question and one we've talked about numerous times. Are there different models that we can utilize for this? Because, again, I mean, putting the viruses together in the lab and testing mice and whatnot, is relatively straightforward, doesn't cost that much money. But when you start upscaling it a thousandfold and producing clinical grade materials, that ends up getting incredibly expensive, and actually running the clinical trials, is limiting for an academic institution the way we've been doing it historically. So, are there avenues by which we can do something in an academic, coming from academia, doing something in an academic kind of model so we don't have to go through uh, very long, drawn out, very expensive clinical trials that the way we're doing it now, because it almost guarantees that you have to have industry involved with pretty deep pockets and venture capitalists and whatnot to be able to make all those things work. So I would like to think that we can come up with some ideas about how this might work, but it's not the standard model that we're currently dealing with, I'd have to say.
Thank you. Erica, as you look forward to your new legislative session, is there something that's happening maybe in another state that you're really excited about that you'd like to maybe see implemented here in Minnesota?
Yeah, that's a great question. You know, you said Minnesotans don't like to brag, and it's so true. We tend to be sort of the leader in the legislative space in these things. But I would say perhaps not another state, but one of the really exciting things that was launched a couple of years ago, and it's just getting off the ground now, is a sickle cell access pilot, and that's actually run by the feds. So the federal government, the Centers for Medicaid, CMMI, the Center for Medicaid Innovation launched a pilot to help state Medicaid programs be able to reimburse for these gene therapies. So that's really exciting. So we're really trying to work with the state of Minnesota to do something like that at the state level. And so that's one of the really exciting things happening. Massachusetts is doing some good work on reimbursement. So definitely looking to some of those states.
Great, thank you. Joey, I'm hoping you can get kind of into the nitty-gritty of the science with us. There was a question on, do you have to change the DNA in every single cell, or how does that work? And I know that each rare disease is kind of different, but someone was just trying to conceptualize what that looks like, if you can help us try to understand.
Yeah. So, in the sense of a lot of these rare diseases that have the, well, as I stated before, cross-corrective capability, you just need to provide kind of a saturating amount because it will traverse throughout the body. So a lot of lysosomal storage disorders are going to be able to take some of these micropharmacy approaches, and we'll hopefully see clinical success moving forward with these. When it comes to the technology to truly create like a saturating um, genome editing strategy, that doesn't really exist yet. There are, different approaches that use lipid nanoparticles to deliver genome editing reagents. So they'll deliver like mRNA that gets transformed into a genome engineering protein, and you can co-deliver DNA templates that can also be delivered at the same time and use like a CRISPR strategy to knock in. But as of now, those aren't completely saturating either because you can create lipid nanoparticles that traffic to like the liver or have certain tissue tropism, but it has yet to be found to a way to completely saturate and hit every single cell in the body. Um, one potential modality would be earlier on kind of hitting the stem cell population within the bone marrow for certain indications of disease. But if it is a, systemic thing that requires full body coverage, I don't necessarily think we are there. But at the individual progenitor cell level, yes, we can change individual nucleotides and correct those errors. Yeah.
Fantastic, thank you. This question could kind of be for anyone on the panel. I want to make sure our listeners know that in the University of Minnesota and at the state of Minnesota, we are really leaders in newborn screening, and that is something that all of our teams have been very passionate about. And like Dan mentioned, when you're dealing with a rare disease, you're facing the clock, and so the earlier you can get the diagnosis, the sooner you can get on it. Does anyone want to just share a little bit what's happening right now in the state of Minnesota as far as newborn screening and what maybe that looks like in the future?
I can
Erica,
take
you
the
want to take that
future
one?
part. I'll take the future part.
Yeah, Paul, why don't you
Okay.
start and then Joey can go.
You want to do the future part? Is that what you said?
What's happening right
I
now? Why don't you
Oh,
start,
well,
Paul, with
so
what's happening right now? Yeah.
Well, so Minnesota's a leader in newborn screening, I think clearly, and we do a tremendous job. And the Department of Health at Minnesota is great. So we're already leaders in this, but I mean, Eric and I have been working on newborn screening for metachromatic leukodystrophy. So I didn't really talk about this in the presentation, but if kids are already affected by the disease and they're already symptomatic, we can't do anything. They progress so quickly and they're just going to die. So we have to, vast majority of the kids that were treated on the clinical trials were kids that were second siblings. So the initial brother or sister was diagnosed because they were symptomatic and they were going to progress and die because nobody can do anything about it. So, but their younger brother or sister, who is yet not symptomatic from the disease, those were the kids that went on those clinical trials. So obviously that's unacceptable, is to have to rely on a system like that. So we need to be able to identify these kids as early as we can so we can effectively impact their disease. And newborn screening is clearly the way forward for some of these kinds of disorders. So we've been working hard to, and Minnesota is going to be one of the first states that's going to start screening for metachromatic leukodystrophy probably in the next few months, I hope. So again, it'll give us the opportunity to see these patients first and intervene while there's still time to do that.
Do you have an idea of how many things are screened currently?
Erica does.
I believe we screen for 64 diseases,
Something like that,
64,
I think,
65. They're getting added, there have been a few additions. So yeah, but in the mid 60s is what we screen for in Minnesota.
I'm sorry, maybe we should just mention, so because people may not recognize this, but when babies are born, before they leave the hospital, they do a heel prick and get a few drops of blood, and it just goes on a card. It's called a Guthrie card, and that goes off to the lab, and you can screen for these 64 diseases. So, again, people that just delivered a baby have other things to think about a lot of times, but that's how that works.
But for Joey, if you don't mind, before you go to the future, and maybe you'll sort of mention how genetic testing is kind of coming into the space. Before that, though, I would be remiss if I didn't say, I really think it is very important for all of us to continue to advocate for newborn screening and the value of newborn screening. Unfortunately, the federal panel committee that recommends what diseases are added federally was dissolved. And so it's really going to be up to the states to make sure they keep their programs strong. So newborn screening is just so fundamental, as Paul said, with so many rare diseases. Once those symptoms shows, it's too late for a treatment. So we need to make sure that we're really prioritizing newborn screening. And fortunately, we live in a state that is a leader. But let's keep that going. And then Joey, you can kind of take us into what the future looks like.
Yeah, let me get off mute. Classic me. So yeah, the one thing I do want to really quickly show is something. This is old data now. It's from 22, but it's the cost of sequencing per human genome going back to 2001. So in 2001, it cost about, this much to sequence the entire human genome, and now it's dropped in 22 to about $1,000 per human genome for whole coverage. I do believe that Illumina in 2024 got the cost down to $200 per human genome. And the idea here is we're able to, through parallel processing and other tricks through the sequencing pipelines, do this at a hugely reduced cost. And with now to bring in AI into this space that can do pattern recognitions and process these large, massive data sets, and the talent we're even bringing in here at the University of Minnesota for bioinformatics, which is the process of taking those giant data sets and breaking them down and finding those patterns. It would not surprise me if there's an explosion in being able to identify what the root cause is for rare genetic disorders and get them, turned around relatively quickly in the future.
Well, that is a great note to end on for hope and future for what that looks like here for the state of Minnesota and beyond. I just want to say thank you to all of our panelists for your time and sharing your stories that impact rare disease and gene therapy. And thank you, everybody, our amazing audience, for your great questions and your engagement in the MIDI Medical School and joining us today. We will follow up with an email, so look out for that. And we really hope to see you next week for our last session, which is going to be focused on cancer. So same time next Tuesday, we're going to speak with another really amazing group of researchers and experts and a nurse to give you a holistic view of what we're doing in the cancer space as well. So thank you, everybody. Have a great evening.